Hello everyone. Today I will present our work on Sort Inspector, an open source framework for multimodal neural data analysis with seizure-specific extensions. Epilepsy affects over 65 million people worldwide. However, its mechanisms remain poorly understood. A major challenge is bridging the gap between microscopic neural activity and macroscopic EEG signals. Spike trains captures millisecond interactions of neurons, while EEG reflects large-scale activity from some local field potentials. If we only rely on EEG, we would miss cellular mechanisms that actually drive seizures. Bridging this micro-macro divide is therefore essential for understanding seizure initiation and propagation. To address this, we developed Sort Inspector, a cross-platform Python framework that unifies Spike, LFP, and EEG analysis in an interactive system with extensions adapted to seizure research. So now, let me walk you through the architecture of Sort Inspector and the single modality functions that serve as its foundation. We start with data input and representation. Sort Inspector accepts outputs from commonly used tools like offline sorter, QSort4, and MATLAB seizure annotations. These inputs are converted into a unified format, ensuring compatibility and consistent workflow. Then, the processing modules compute neuron properties, local connectivity, spike field relationships, and track changes across EEG-defined seizure stages. All of these are accessible through an interactive graphical interface with single-click execution for quantification, visualization, and exporting results. This lowers the barrier for users who may not be expert programmers. Now, looking at the single modality analysis. First, neural identity clustering classifies neurons into regular or fast spiking subtypes using waveform features, with support for template loading, manual editing, and clustering algorithms. Second, signal displays and frequency analysis provides one-click visualization of EEG, LFP, and spikes, with standard spectral and firing rate analysis. Third, channel map analysis visualizes electrogeometry and channel cluster relationships, both in an interactive spatial map and in tabular summaries. Fourth, seizure segmentation applies a Tigre energy operator to EEG signals, detecting frequency shifts and finer seizure stages. This provides a temporal framework to align to align spikes with seizure dynamics. And finally, the PSTH analysis evaluates firing rate dynamics in multiple configurations. This includes single cluster overlays, grouped by cell types, or even across experiments. Building on the single modality foundations, Sort Inspector integrates modalities to uncover cross-scale relationships. First, we have EEG-guided monosynaptic connectivity inference. Using the Connect algorithm, we infer monosynaptic connectivity and postsynaptic potentials, visualized as matrices, heat maps, graphs, and even seizure-aligned views. Based on this connectivity results, we then implement pairwise and triplet motive analysis. Pairwise motives classifies neuron pairs as excitatory, inhibitory, or mixed, and triplet motives extend this into 13 canonical triads and sign variants, quantifying network structure across different conditions. Then we provide modules for spike field relationships. Firstly, the spike triggered average computes the mean LFP waveform aligned to spikes in either trial or seizure modes. Secondly, the field triggered average then aligns spikes with oscillatory phases of a filtered LFP, producing histograms and polar plots of spike phase relationships. Thirdly, the spike field coherence analysis quantifies frequency domain coupling between spikes and LFPs using sliding window FFTs. Outputs include spectra, significance thresholds, and phase relationships, allowing us to assess the synchrony. To summarize, Sort Inspector is an open source cross-platform framework that unifies spikes, LFPs, and EEG analysis into a single interactive interface. It provides powerful single modality tools and extends to multimodality modules that bridge the micro-macro divide critical to seizure research. By doing so, we hope to enable more mechanistic insights into seizure initiation and propagation, and ultimately support the development of better intervention technologies. Finally, I want to acknowledge our funding support. This work was supported in part by the NIH grants, with additional support from a Carnegie Mellon SURF Fellowship. Thank you very much. That's my presentation.